Thank you, thank you. So today we're just gonna do, we're gonna do two sessions. We'll see if we can get these knocked out so we don't run over. I know yesterday we were able to get out a little bit earlier. I wanted to make up for the day before that we ran a bit over. So today we're gonna talk about bioinformatics. In the first little set of slides, it's about 25 slides. And then the second set, we'll talk about the regulation of genetic testing, how genetic testing is regulated from a commercial perspective, and kind of what's involved in that. So some of the stuff in this introduction to bioinformatics is going to be kind of a carryover from yesterday's lecture on genetics. I had put the two topics kind of together and put some of the basic details in both talks. So some of the stuff that we already talked about in the lecture yesterday when we talked about genes, particularly on Monday, is kind of covered in here again. So I'll gloss over some of that stuff. Again, if you have any questions, please just go ahead and say something. I can't see the viewer panel right now, so I can see more of the slides. So if you got a question, please just unmute yourself and say something. But we'll go ahead and get started right now. So this is going to be a discussion on bioinformatics. There'll be a little bit of a discussion on proteomics as well. We'll explain what those are, and just to kind of go over briefly what bioinformatics is, what are some of the applications, and how these affect science and research going forward. So just kind of some terms again to make sure that you're familiar with, and we've talked about most of these over the last couple days, so none of this should be really new. We will go into a little bit more detail. I know we talked a bit on Monday about how proteins are put together, and when we get to proteomics, we'll talk a little bit more about protein structure and kind of how that all works together. But here's some basic terms, and I think again we've kind of gone over these pretty well the last few days. So again, just to kind of remind everybody, and I didn't beat you to death with this too much over the last couple days, but there's a term that we use that's called the dogma of biology, and this basically describes how we see information in biology flowing. And when we talk about information in biology, we're primarily talking about genetic information. And this is the classical paradigm, and many of you have probably seen this, that DNA makes protein, and then protein makes a function. So you have this flow in biology from where we have a DNA strand that's going to be inside of every nucleated cell. And then this DNA strand is going to encode for proteins. And then the expression of these proteins will carry out some function within the cell or within the organism. It might be the expression of a protein that's going to be used to stimulate an immune response. It could be the expression of a protein that's used to help rebuild your hair or your nails. But all this information comes from DNA. So we have this flow of information in biology that starts at the DNA molecule, which produces a protein, and then the protein is going to end up carrying out some kind of a function. So we can sometimes refer to this as kind of a top-down or a bottom-up effect. So if you look at this graph here, we see that we have animal species, and within those species are specific organisms. So let's say, for example, the species is Canis lupus. This is going to be dogs and wolves, or this is going to be the canine species. And then within there, you've got different types of dogs. You've got wolves, you've got poodles, you've got German shepherds. And then comprising all of those different types of dogs within that general heading of species are all the cells that make up each one of these organisms. So you have a species. The species group is comprised of multiple different organisms, but all those organisms are going to be made up of a cell. Inside of the cell, the cells that are nucleated, most of the cells are going to have a nucleus, which means they're going to have chromosomes, which means there's going to be DNA present. And then we talked about this over the last couple of days. Within the DNA, the functional unit of DNA, we call these genes. And then these genes are themselves going to be comprised of base pairs. And this is the A's, the T's, and the C's, and the G's that we talked about for classical DNA base pairing that comprises the whole entirety of the DNA strand. From these bases, remember we talked about the codon usage table, how 3 bases, an ATC or a GGA or a UUG, will encode for an amino acid, and those amino acids are going to be brought over and they are going to make up the protein structure itself. So this is kind of a substructure diagram, kind of showing how, this substructure effect works at the species level all the way down to the amino acid level. And again, all of this information that we're getting from here is coming from the DNA strand itself. And again, just to kind of remind everybody, the genes are going to help make these amino acids, which are then going to fold into the proteins. The proteins are going to have some kind of a function which will affect the cell. This in turn will affect the behavior of the organism. Maybe you're having to produce a lot of protein that's going to help you more efficiently use glucose because you're running for your life across the savannah from a herd of angry lions, from a pride of angry lions. And so you need your cells to rapidly make these amino acids so they can comprise these proteins. So these proteins can be used to help efficiently shuttle glucose in and out of your cell so your cells can respire, can process oxygen and other metabolites efficiently so you can run faster and hopefully avoid being dinner. Oh, I'm sorry. So just to kind of remind everybody again on what the cell is, the basic unit of life, we know that cells have all of these different little substructures within the cell that carry out different functions. Obviously, from looking at the picture of this cell right here, we can tell that this is a plant cell because it has chloroplasts. And the chloroplasts are the structure that plants use to carry out photosynthesis. This is kind of basic biology 101. So the cell is our basic unit of life. We have different types of cells depending upon where they are in the body. They're all going to have different biological functions. Cells make other cells. This is part of the cell theory of life, the basic theory that describes life on Earth. One of the tenets of that theory is that cells come from other cells. So cells just don't spontaneously come out of nowhere. They're actually produced from other cells. And we get cells usually produced by other cells from using two mechanisms. One mechanism can be mitosis. This is just going to be cell division, where the cell basically makes a copy of itself. And then we can also have meiosis, which is where we have the combination of two cells kind of coming together and forming this daughter cell that's going to come off that can be used to then make other cells. And so we see meiosis particularly occurring when we have sexual reproduction. When we have the fusion of the sperm and the egg, we're going to have this mitotic event occurring where these two things are going to come together. And this is the classical thing we've always talked about in biology, where we have the different phases of cell replication. This is the prophase, the metaphase, the anaphase, the telophase, all these little cell divisions categories that many students are required to memorize. And remember, during metaphase, you're going to have the chromosomes condensed. They're going to align in the center of the cell, and these two cells that are fused together that are exchanging chromosomes in the middle. Some of the when they split apart, they're going to take chromosomes away from each other, and this is how we generate genetic variability within organisms. So we have cells making other cells. We're going to have either mitosis or meiosis producing new cells. And then just kind of keeping in mind when we talk about the cells that we're referring to here that have a nucleus, we're talking about primarily eukaryotic cells. Eukaryotes are us, are mammals, or most animals. Prokaryotes are going to be things like bacteria, for example, that don't have a nucleus. So kind of just reorient everybody on what the basic structure for the cell is. Of course, every cell is going to have a nucleus, and inside of them are going to be the chromosomes. We have different pairs of chromosomes that are going to be linked up. You can see the chromosomes, we number them from 1 to 22 in humans, for example. So we have the chromosomes, the general nomenclature is the smaller the number, the larger the chromosome is. So you can see chromosome 1, for example, is really big. 2 is almost as big. 3 is not quite as big. And you can see the descending order in size as we go down the numbering system, such that when you get to chromosomes 20, 21, and
How are
22,
you? Good, thanks.
they're very small. And then we also have, for example, in humans, we have the X and the Y chromosomes. These are generally referred to as the sex chromosomes. Females are going to have 2 X chromosomes. 1 X comes from your mother. 1 X comes from the father. In the case of men, they're going to have X and Y. The X chromosome comes from your mother, and the Y chromosome comes from your dad. And then there's going to be different numbers of chromosomes depending on what species you look at. In humans, for example, we have 46. In the tobacco plant, 48. Goldfish and chimp have different numbers. And these chromosomes are usually going to exist in pairs. So if you look at chromosome 11, for example, it's actually going to be two chromosomes that comprise chromosome number 11. And then, as I said, and this is a paradigm that's pretty much consistent across all forms of life we've looked at on our planet. We delineate and identify males as being XY and females as being XX. Now, this is different in birds. Birds are the one exception to the rule. In birds, males are XX and the females are XY. But beyond that exception, everything else is defined by XY for male and XX for female. So just kind of to recapitulate again, the chromosomes and the basic structure of the cells itself. We kind of talked about DNA already, that every cell, every nucleated cell in your body is going to have DNA. The DNA is supercoiled. I didn't really mention this in the past, but if you were to take out the DNA in one cell and unwind it and stretch it out, it would actually stretch out to be about 6 feet long. So you have to ask yourself, how do you take something that's 6 feet long and condense it and pack it down to where it is so small you can't see it with your naked eye? None of us, as far as I know, can see cells with our naked eyes. That's why we invented microscopes, so we could be able to see cells. The way that this occurs is through a process that's called supercoiling, where basically the DNA molecule is coiled so tightly and it's so condensed that it condenses down into a size that is not visible with the naked human eye. But again, if we could take the DNA molecule out of a single cell and uncoil it and stretch it out, it would be about 6 feet long. And just as we've talked about previously, every cell, nucleated cell, is going to have a DNA molecule inside of it. We talked a little bit about the Human Genome Project, about how we initially sequenced the human genome in the mid-90s. It took us 13 years to sequence the Human Genome Project about 25 years ago, or I guess it's been about 30 years ago now. And we're now at the point to where here at the Center for Genomic Research and in the Department of Human Genetics, we can sequence a genome in about 6 to 8 hours. And of course, we've done genomes for many organisms on the planet. The fruit fly, obviously, I don't know how many of y'all did fruit fly genetics labs if you've taken genetics, but the fruit fly has been heavily sequenced. Of course, we've sequenced mouse DNA because we use mice a lot in research. And then other things like, and this is not a complete list, we've sequenced dog genomes, chickens, many of the bacteria, exotic things like squid and pufferfish. Pretty much anything that we've been able to extract DNA from, we've gone ahead and tried to sequence the genome. And so we've collected a massive amount of information over the last 20 or 30 years from all these genomes. And one of the issues we're trying to confront now is how do we interpret all of this DNA? Just to kind of remind everybody again, the DNA molecule is a double helix. We have the base pairing, the A's and the T's are pairing with the C's and the G's, and that's kind of shown on this little graph right here. Remember, all these base pairs are held together by very weak hydrogen bonds. And so when we want to replicate this molecule, all we need to do is input a little bit of energy to break these hydrogen bonds. And then once these hydrogen bonds are broken, the helix can spread apart. And then we start to get DNA replication occurring on each one of those strands. So if we look at differences in DNA and we kind of look between the species and stuff, we can use DNA to help us differentiate between individual animal species, right? Because our DNA is not going to be the same as a mouse or as a banana or as a chimpanzee. So we can use differences in DNA to differentiate between species. Within those species, we can use special markers within the DNA to differentiate between different members of the species. So we can not only differentiate between gender, but we can also tell the difference between a rhesus macaque and a Sudi mangabe. They're both primates, but they're different and they have different DNA. And then we can also use this information to differentiate between individuals themselves. And so this has kind of formed the basis for how we sequence and interpret DNA today. Of course, we share a lot of DNA. We've talked about it, and you've probably heard this many times. You know, there's only about a 2 to 5% difference. It's actually closer to 2% difference between us and non-human primates. Only about a 2% difference. The difference between, you know, Joe Biden and Whoopi Goldberg is very, very tiny. It's really going to come down to small differences that are going to be on the DNA molecule that are going to code for things like maybe differences in hair color or eye color or height or weight or muscle structure. but we're going to have very much the same genes in terms of the genes that make arms, the genes that make legs, the genes that form our eyes and our brain structure and our central nervous system. So very, very small differences between individuals within a species. We actually do share some genetic material with plants, for example. We think that the way that we've seen a lot of genetic material kind of move back and forth amongst the different species on our planet has probably been through viruses. Some viruses like the HIV virus are able to integrate themselves into the genome of the individual that's been infected with the HIV virus. And HIV actually puts its viral genome inside of the host genome where it sits there for a while. And then when the virus is ready to leave the host and go and infect a new host, it takes its DNA out of the host and packages it to go and infect a new host. Well, when the HIV virus takes its DNA out of the host, it's going to take some of the host DNA with it. So when you have HIV, for example, which is categorized as a retrovirus, we have a lot of retroviruses on this planet that seem to do the same thing. They infect a host, they put their genome in that host, they integrate their genome into that host, and then when they decide to leave that host and go to a new host, they have to take their DNA back out of the host. Well, it's not a clean procedure. So when the HIV virus takes its DNA out of the host, it's going to take some host DNA with it. And then it's therefore going to transfer some of that host DNA to the next host. And so this may be one mechanism that we see in our planet that helps keep genetic diversity high by having viruses with the ability to infect multiple different hosts and kind of pull DNA from one host to the other. I know very commonly we talk about where did HIV come from? And there's a big joke about HIV came from a hunter having sex with a monkey. I think I've heard that thing a million times since the 1980s, because if we look at the HIV virus, there is a very similar virus in monkeys called simian immunodeficiency virus. So HIV stands for human immunodeficiency virus. There's also one that infects monkeys. It's called SIV. And what's more likely to have occurred, and as somebody that's done work with non-human primates, it's almost comical to think that somebody would prescribe to the notion that some dude out in the forest captured and had sex with a monkey, and that's where HIV came from. If you get close enough to a monkey where they can touch you, they're probably going to rip your arms off and beat you to death with them. So the simple concept of somebody romancing a monkey to make HIV is completely outlandish. But what's more than likely that happened is that monkey is very commonly eaten overseas as a meat product. I spent 2 years in the Philippines when I was in the Marines, and I ate a ton of monkey. It's very common to eat over there. And what's more likely to have occurred is that somebody was out hunting in the forest and they killed a monkey that had an infection with simian immunodeficiency virus. They probably didn't cook the meat well enough. They ate the monkey meat and they got the virus. We see the same mechanism happen with virus infections all the time. If you remember, this was one of the theories that they put forth in early 2020 for how the COVID pandemic began. Remember, it came from the wet market that's right next to the Virology Institute. Somebody went over there and ate meat from the wet market. And we do have confirmation that the Wuhan Institute of Virology was taking their extra animals and selling them to the wet market as food. So they would do an experiment, and if any of the animals survived, they would take them over there and sell them to the wet market for food. So it could make sense that somebody ate, that they did a challenge experiment where they infected a bunch of monkeys with COVID. They had some survivors, and after a year, they're like, well, what do we do with these things? We're sick of taking care of them. Well, take them over to the market and sell them. So it would be, it wouldn't be a big leap to think that maybe the pandemic began from an action such as that, because we do know, based on the records that we were able to get before China quit sharing information with us in 2020, that they did have monkeys and animals from that civets and Pseudomangabes, which is one of the models used for coronavirus animal challenges. We do know that some of the staff from that Wuhan Institute of Virology took those animals over there and sold them at the wet market. So it's not inconceivable that you could consume infected meat and then get a viral infection. We've actually done studies here where we've done a lot of work with Zika virus in using animal models. And one thing that we've noticed is that we use a possum model down here to infect with Zika virus. Not the same possums that you see running around outside in the street. These are possums that are about the size of a mouse that are from Brazil. They're called the short gray-tailed opossum. And we've used them very heavily in the last 5 years to study how Zika virus infection works, because these animals are susceptible to Zika, but they have a really strong immune system, very similar to human beings. What we've seen is that when we infect the pups with Zika virus and then give them back to the mother, sometimes the mother will eat the infected pup, and then the mother comes down with Zika virus from consuming an infected pup. So the transfer of viruses through consumption of infected substances such as meat is not an uncommon thing at all. So it's probably much more likely that HIV was introduced into the human population from somebody eating infected monkey meat. But because there's such a small difference in the genetic material, it's large in the sense that if you look at this and say, well, there's only 4% difference between Joe Biden and this mouse, you might argue that the mouse is probably beating Joe Biden here, but you say, well, 4% difference, shouldn't the mouse look more like a human being? But that 4% difference is what gives us this organism compared to this organism over here. Excuse me. And even between non-human primates, we're talking about a 2 to 3% difference. And so these small differences probably have helped facilitate viral infections across species, because many viruses are able to adapt to cells that they infect. And we've been able to show experimentally that if we take a virus that only infects monkeys and we grow that virus with human cells over and over and over, you can eventually adapt that virus to grow and replicate and infect human cells. This is how we got the COVID pandemic. That SARS-CoV-2 coronavirus was made in a lab in Wuhan in 2014. Dr. Anthony Fauci and the researchers that did this published papers in 2014 and 2015 showing that they had made this virus. What were one of the steps that they did to make this virus? They took the original coronavirus isolate that they had gotten from bats and grew that bat virus with human cells for about a year. And over time, the virus from bats adapted to start to grow in human cells. This is highly illegal, by the way. This is part of that gain of function stuff that we had made illegal in this country, but Fauci got around it by having the work done in China. And this is where we got the COVID pandemic from. So small differences in DNA can still allow for viruses to jump between species. And what you're going to get as a result of it is going to be kind of up to you. But I think in general, having these small differences in DNA between species is a benefit to us because it helps keep genetic diversity, fresh and somewhat novel. Again, we talked a little bit about genes. I think we put this actual slide up, or this picture. Just remembering again that within the chromosome, the functional unit is the genes. Most of our DNA, about 95%, we refer to as the junk DNA. These are the introns, the stuff that is just kind of hanging out there that we don't really know what it does. And then we have about 5% of our DNA that's actually expressed. These are called the exons. Remember the exons and the introns. The way I remember it is exons are expressed, EX expressed, EX for exons. The introns are what separate the exons, and the introns are inside and they're not expressed. So again, just to kind of remind everybody as well, once we get our DNA made, the DNA is going to be converted and transcribed into messenger RNA, and then the messenger RNA is going to be translated into protein. So we have unwinding of the DNA molecule, we get transcription that makes RNA. And so here we have our DNA flowing into RNA. The RNA is eventually going to make its way over to the ribosome. We're going to get translation occurring. All those little amino acids are going to come in and get plugged in. And this is what's going to form our proteins. So again, just to kind of put this up here so you can look at this at your own time if you want to, but we've talked a bit about this already with how the transcription part works. And remember, in the RNA molecule, there's no T, there's no thymine, so it's always uracil. So the easy way when you ever look at a DNA molecule and you're not sure if it's DNA or RNA, you look for the presence of U's. If you see a U, you know it's an RNA molecule. If you don't see a U in there, then it must be a DNA molecule. And eventually, as I said, we get unwinding of the DNA, transcription to make RNA, and then the RNA is going to be translated to form the proteins hooked up with the different amino acids, ultimately give us our protein that's going to hopefully be able to go out there and carry out its function within the cell. We've already talked a little bit about the genetic code, just to kind of remind everybody about the codons again. We already looked at this codon table again. We did some example synthesis stuff, just kind of reminding everybody that the codon is read in triplicate, so 3 bases encode for an amino acid. And remember, this codon has the ability to wobble at this 3rd base, at this very last base. That's how we get a bit of a redundancy. We call this redundancy in the genetic code. This kind of gives us some flexibility in case there is a mistake during translation, and maybe CCU is changed to CCC or CCA instead. Well, guess what? You're still going to get a proline amino acid. So there's some flexibility within the genetic code to tolerate for problems. So kind of getting into what the main rest of the lecture is going to be talking about as we kind of shift into proteomics is kind of looking again at proteins. So remember, the proteins are coming from the DNA molecule, which is telling the body or the cell what amino acids to hook together in what order to make the protein. And eventually, all these amino acids are going to be hooked up together, and we're going to get this protein molecule. It's going to fold into a 3-dimensional shape. And remember, I talked about protein's function is determined by its shape. If the protein does not adopt the proper shape, it will not have the proper function. So we always talk about in proteomics and in protein discussions, shape determines function. If the protein doesn't have the proper shape, it's not going to have the proper function. And within this protein molecule, you can have different types of folds. You can have different types of structures. Some of these structures can be very resistant to water. Some of these structures within the protein can be very dissolvable in water. So you have parts of the protein that are hydrophilic and parts of it that are hydrophobic. If you probably remember from basic biology, when we talk about something being hydrophobic, that means it's water hating. Phobic, phobia, fear. So hydrophobic means that a substance is water hating or fears water. Hydrophilic means that the substance is water loving, which basically comes back to when something's hydrophobic, it's not going to dissolve very well in water. If something's hydrophilic, it's going to dissolve very well in water. And proteins are always going to have part of the molecule that's water hating, that's hydrophobic, and part of it that's hydrophilic. When a protein is made, it's always going to fold into the same conformation. If for any reason a protein gets made and it doesn't fold into the proper conformation, there is an organelle, a substructure within the cell that's called the proteasome, that can basically break down the protein and recycle the amino acids so the components of the protein get reused. So many times when proteins are used within the body and they either carry out their function and they're no longer needed, or the protein was made improperly, all proteins in the body eventually get broken down through this structure called the proteasome. And once they're broken down, they just get converted back into the amino acids. So the building blocks are there to be used again. This is analogous to if you've got bricks in a wall outside and you want to build a new wall and you don't need your old wall anymore, rather than going out and buying a bunch of new bricks, maybe you can go out there, carefully knock down your old wall, save as many of the bricks as possible, and use them to build your new wall. This is exactly what the body is trying to do so it can conserve energy and reuse resources as much as possible. So, and I know we talked a little bit about this again in terms of how inheritance is caused, and just to kind of remind everybody, when we talk about how we get genetic variability within an organism, there's really 3 ways that you can change or make changes to your DNA. One is that you're going to inherit new changes. So your mother and your father, half of your DNA is coming from your mom, half is coming from your dad. Is it exactly half? It's not. It can change a little bit, but just for the sake of discussion and for the purposes of teaching, we usually talk about half comes from mom, half comes from your father. This is primarily based on the fact that you do get one chromosome from your father and one chromosome from your mother. This is where we get the term mama's boy and daddy's girl. Women have two X chromosomes. They'll get one X from their mother, but they'll get the other X from their father. The X chromosome contains most of the information that's going to be expressed in the organism. So if you're getting the X from your mom and the X from your father, and you've got those two X's, one of those X's is always active. Two X chromosomes cannot be actively used to express proteins at the same time. So this refers to something y'all may have heard of called the lion hypothesis. And this is based off of looking at calico cats, the cats that have 14 different colors on them, the different patches. That coloring, if you look at calico cats, almost all calico cats are female. And the reason that they have that different coloring is that sometimes in their life, one X chromosome is turned on and they get genes expressed from that X, and then that one gets shut off and the other X chromosome gets turned on and genes are expressed from that one. So you get this variation in the cat's color design, for example. So women are getting an X from dad and an X from mom. And so sometimes in that woman, the X chromosome from her father is going to be expressed. And so she's going to be displaying genetic characteristics from her dad. In the man, we only get one X chromosome and it always comes from mom. So most of the genetic expression that's happening in the man is going to be based on the DNA that he got from his mom. The Y chromosome comes from the father to the son. The main component, based on the knowledge we have right now, is that most of the stuff that's on the Y chromosome are sex genes, the genes that encode the penis and other organisms related to sexual reproduction in the man. So for men, a lot of the gene expression is going to be off that X chromosome. And since we only get one, our X is always active. So this kind of leads to the term of mama's boy and daddy's girl. And there is kind of some genetic basis in this when you look at how genes are inherited from the parents. So besides inheriting genes, you can, I'm sorry, was there a question?
Dr. Thomas,
Yes.
can you allow Christina into the room? I think she's waiting.
Yeah, did you hear that, Wanda?
Yeah, I got it.
Okay, thank you. So you have the ability to inherit these genes from your parents, and then depending on what your sex is, you'll get expression from these chromosomes at different points in your lifespan. You can also have changes to your genes from mutational events. These mutation events could be exposure to certain chemicals, could be exposure to radiation. It could be exposure to a virus like HIV that maybe, or another retrovirus that comes in, and when it comes in and infects you and inserts its genome into your DNA, it's also bringing with it DNA that it had from its last host. This can cause a mutation. And you can also get what are called crossover events. And this is when, and again, if you think back a few slides ago when we were talking about mitosis and meiosis, there's going to be a point during that cell division cycle where all of the chromosomes are going to align in the middle of the cell along what we call the metaphasic plate. So you have these two cells that are fused, they're coming together, and at the middle of that fusion, there's a line, an imaginary line there, and that's where all the chromosomes from both the cells are going to be pushed and they're going to line up. And those chromosomes actually physically touch each other in the cell. And when they touch each other, they exchange DNA. And then eventually those chromosomes are pulled back to the cells that they came from. But when they're pulled back, they're different now. So we refer to these as crossover events. So you have inheritance, mutation, and crossover that are really used to provide the most genetic variability that we see within a given species. And genes can also change slightly just as a normal process of replication during the life cycle of an organism. One of the things we've noticed in the last 20 or so years is that we are starting to see increases in the rates of cancer as people get older. And if you think about this from a genetic perspective, there may be a bit of sense to this phenomenon. So the longer that you live, the more that your DNA is being replicated. Remember, we've got about 6 billion cells in our body, and anywhere from probably 1 to 2 to maybe 3 billion cells a day are being replicated. So we're making new cells, billions of new cells every day. Billions of times the DNA molecule is being copied. There's a lot of opportunity for mutations, for changes to occur. They could occur from maybe you're under a lot of physical stress and the cells feel that physical stress, and maybe some of those stress events causes the enzymes that do DNA replication to not function quite as well as they would, and maybe they make a mistake. And again, I've told you that we have a mechanism within the body to go back and correct changes or mistakes that are made in DNA. But that doesn't always happen. Sometimes the system just isn't, we're not a perfect organism. The system isn't perfect. And sometimes mistakes, even if they are identified, don't always get corrected and they get carried on instead. Sometimes when genes are copied, a base might be deleted, an extra base might get inserted, maybe the entire codon gets changed for some reason. And so we can have mutations arise because a base got deleted. Maybe this should have been a C, but it gets changed into a G for some reason. Maybe this right here got an extra letter inserted in. Maybe this T instead gets substituted with an A. Maybe you have a base, this extra T here that gets deleted. Whenever you have changes to the code in here, particularly if you have a deletion, remember when the DNA molecule is being read to make protein, we're reading this in groups of 3. So these 3 are going to code for an amino acid, and then these 3 are going to code for an amino acid. Well, if I suddenly take one of these bases out, then ACG will still code for the first amino acid, but now instead of having TTG, we're going to have TGA. This is going to be a different amino acid. So a deletion can really alter the makeup of a protein. And if we have one base deleted that changes the amino acid profile, there's a very, very high probability that the protein is not going to be made in the proper shape. If it's not made the proper shape, it won't fold the proper way, and it won't have the proper function. So in human beings, mutations are almost always bad. They're almost always deleterious. We don't see mutations happening like we do on TV where you turn into Spider-Man or the Hulk or some crap like that. Mutations in humans are almost always bad. In most organisms, they're bad. There's really only probably 2 organisms, and I use that term very loosely, that I can think of that would maybe benefit from a mutation, and those are viruses and bacteria. Because from their perspective, a mutation can very oftentimes confer an advantage in replication. And so for viruses and bacteria, if there's a mutation, then that virus strain just dies out. But there's many other viruses alive in that body that may not have that mutation that can move on. But sometimes mutations occur that make the bacteria or the virus better able to avoid the immune system. Maybe they're able to replicate a little bit faster. And so in us, while mutations are almost always a bad thing, particularly because of the complexity that we have as a large organism, in viruses and bacteria, mutations half the times are going to be beneficial because it will change their protein structure, and that change may give them an advantage in replication or an advantage in avoiding the immune system that doesn't really work for us. So I already mentioned a little bit about the crossover stuff. I'll let y'all look at this on your own time if you want to. This just kind of goes through how these crossover events can occur and what are some of the different possibilities you can get from a crossover. But if you just look here, just with the mother and the father pairing right here with these 2 chromosomes, these crossover events can generate at least 4 different chromosome variation pairs in the children. And if then you imagine that these children have children and they have similar crossover events, you can very quickly see how DNA can change in just a few generations from the parents to the offspring to the grandparents and so on. So this is another mechanism to kind of help give us some genetic variability within our species. One thing that we've noticed is we've seen animal species on this planet that start to die out or get to really low numbers is that once a species gets to a certain threshold, they lose genetic variability. And at that point, more than likely the species is going to die out. This is something that we've been seeing very happen out in real time with tigers in the wild. Tigers in the wild have gone from being very, very high numbers of tigers to only now a few thousand. And when they sequence the DNA of those tigers, they're almost always very closely genetically related. And we, just as we push against incest, against mothers and parents and children mating together, because from a genetic perspective, for us in particular, not to mention the social stigmas that come along with it, just from a genetic perspective, those offspring don't end up being, very having a very high level of genetic integrity. The same thing happens in animal species. And so this is something that we keep an eye on when we see animals getting down to just a few thousand members of the population in the wild. They're going to lose a lot of that genetic variability. And if a disease or something pops up that those animals can't respond to because they have very limited genetic variability, it's going to end up killing them off. So, but we can use our understanding of genes and how genes are passed down to begin to look at a species over time to make what's called a phylogenetic tree. And this is one of the first steps really that we see when we try to apply bioinformatics using DNA to study how information is flowing. This is what's given us a lot of insight in the last 20 years as to where we've come from. We've always held this common theory that humans came out of Africa, right? We came out of Africa sometime last couple hundred thousand years. A big group of people were living in Africa. They said, man, it sucks here. Let's spread out. And they left and they spread out all over the planet. That's been the traditional mainstream idea for how human beings spread across this planet over the last 400,000 or 500,000 years, is that we all lived in Africa, and then one day we said, screw it, we're sick of this, and groups started leaving. What we've now been able to show using genetics is that we didn't just leave Africa one time. There have been probably a dozen events over the last 500,000 years where large groups of humans left Africa, spread to different parts, and then came back to Africa. And then some other people left and spread out and came back to Africa. Something that was very interesting that was done about 5 years ago, that's a published study, is they went and sequenced DNA from some of these isolated tribes in the Amazon that have had very little outside contact. And certainly from a genetics perspective, they're a very closed population. You don't see members of these tribes, these isolated tribes, leaving to go get girlfriends or boyfriends in the city and then bringing their new, family back to the tribe. They stay within the tribe and breed and marry within the tribe. So they went and did DNA sequencing of some of these isolated tribes to kind of see what their differences in their DNA are between us. And one thing that they found is that there is a tribe in the central Amazonian basin that is very closely genetically related to the Aborigines in Australia. So using phylogenetics, they were able to build a tree that shows that sometime about 40,000 years ago, somebody came from Australia to Central America, to the Amazon, and stayed there because their DNA is there, because the tribes that are in that area have these genetic markers that we don't see anywhere else in any other humans in the planet except for Aborigines that are from Australia. According to the Aborigines, they've been in Australia for, over 100,000 years. And we know from studying their genetics that probably about 80 or 90,000 years, they've been there. How did their DNA, this is one of the big questions that they're trying to grip with right now, is they're trying to explain, how did their DNA 40,000 years ago leave Australia and end up in the Amazon? Because according to modern archaeology, no one was traveling around the world in boats. There were no ships. We were all hunter-gatherers roaming around, in our fur dresses, eating plants and stuff. But yet the DNA data shows that somebody came from Australia 40,000 years ago and set up shop in the Amazon because their DNA was left in all those tribes that are there. So we look at DNA sequences to track these kind of events, to kind of see who evolved from who. We can look at very specific genes. There are certain genes that we refer to as being conserved. That means that these genes are maintained within all of the population members within a given species. So for humans, for example, and this would kind of make sense, the gene that we use to produce hemoglobin, which is the protein that carries oxygen around to all of our cells in our body, the hemoglobin gene in you is the same as me, is the same as you, is the same as somebody from Africa, somebody from, 10,000 years ago. Hemoglobin is a critical protein. And so mutations are not allowed to occur in the hemoglobin gene. And if you do get a mutation there, it's probably going to occur when you're an infant or you're a fetus and you're going to be aborted, because if you have a mutated hemoglobin gene, you're not going to survive. So the hemoglobin gene is a good example of a gene that's been very highly conserved. So we can look for the presence of that hemoglobin gene across many different members of the human species to see, is that gene still intact? Does it still look the same? And this allows us to then put together these trees, these phylogenetic trees, to try to track relationships between different types of organisms. One thing that we have been able to gather from the DNA studies is this concept of LUCA. And LUCA stands for the last universal common ancestor. And what this essentially means is that, and there's a lot of controversy around this, is that, but basically, we can look back at DNA sequences and kind of walking back in time, which we can do as we sequence DNA from different organisms around the planet. We can kind of track and look to see where we started to kind of diverge from the other organisms on the planet. So remember, we share about 2% of our DNA with monkeys, but monkeys aren't driving cars and we're not swinging from the trees in the jungle. So at some point, the DNA that made up both monkeys and humans split off, and one line went to become non-human primates. The other line went on to become Homo erectus and Neanderthalis, and then eventually, Florensis, and then eventually Homo sapiens. We look at this and call this the last universal common ancestor. What was the last organism that existed that shared DNA that virtually every organism on the planet has? And it looks like this LUCA existed about 4 billion years ago, which is roughly when we think life started to really evolve on this planet. Again, there's a lot of controversy in this, but you do see the term LUCA used quite a bit because this kind of refers back to that original DNA sequence before it started to diverge and be spread amongst the different species on our planet. And we refer to this as LUCA. So by looking at the DNA sequences, we can start to put together these phylogenetic trees, which kind of helps us start to getting our hands around this DNA. A lot of what has driven genetic research has been our desire to cure diseases. And we know that, certain genes are going to be more prone to mutation, or if the gene is mutated, that it will cause a very specific disease. And so a lot of our focus in looking at DNA sequencing has been to try to address genetic disorders and the diseases that are caused by them. Different gene mutations cause different problems. So just to kind of give you an example, and this is one that probably a lot of people have heard about, is Down syndrome. Down syndrome is caused by having an extra chromosome 21. So if you think back to the figure that I showed you, the chromosomes all exist in pairs. We have 1 to 22. The chromosomes are very big, but there's a pair of them. When you get down to 21 or 22, the chromosomes are very tiny. Sometimes when the replication process is occurring during mitosis and the chromosomes are aligned on that metaphasic plate and then they're pulled back to their cell, sometimes when they're pulled, they don't get pulled all the way and one gets left behind. Oftentimes, if one is left behind, it's going to be chromosome 21. So what ends up happening is you end up having a cell that has not 2 chromosome 21s, but 3 chromosome 21s. And then that cell that has 3 chromosome 21s is now going to start to go through cell division, and it's going to make more cells with 3 chromosome 21s. And having this extra chromosome leads to some different mutations and different altered functions of proteins. And this is the classical genetic marker that we see associated with Down syndrome. Cystic fibrosis is another genetic disorder that's caused by a mutation, and it's a single base pair mutation. So one of those base pairs, the A, the T, the C's, and the G's, one of them gets mutated, just one. Out of the 3 billion that we have in our genetic and our DNA molecule, one gets changed, and that one mutation occurs in an area that encodes for a protein that's used to help your lung cells allow ions to move in and out of the cell. If the flow of ions is altered coming in and out of a cell, this is going to have a dramatic impact on not only how the cell functions, but the flow of ions inside and outside of cells changes the electrical field of that cell. And when that change occurs, that change in the electrical field of the cell is going to then cause the cell to restrict other things from coming in. And so things that the cell should normally be able to ingest or bring in now can't come in because of that single base pair mutation that's disabled the protein that's used to regulate how ions come inside of lung cells. And if those lung cells can't proper functionally, they are not able to move fluid outside of the cell. And if fluids can't be moved outside of the cell when they need to be moved out in order to maintain balance within the lung cells, then you're going to have problems with those cells. They're going to be soggy, and this leads to cystic fibrosis, which is severe breathing problems that are associated with this single base pair mutation. So genetic disorders are really driven a lot of our desire to understand how DNA works. And because we now know that the proteins coming from DNA are so important, this is what's really led to this approach now that we call bioinformatics and proteomics, which is really trying to study how the genetic trees lead to the formation of certain proteins and how then this altered protein structure can perhaps lead to disease. So if the protein gets mutated in any way, then the protein shape is going to be changed. And remember, the shape determines the function. So if we lose this function, then we're losing that protein. One of the things that we've been trying to do, and it's often referred to as the holy grail of bioinformatics, is to figure out, is there a set of rules for how proteins fold? Because you would, and there probably is to some extent, but this is something we have not been able to figure out yet. What tells a protein that has a certain amino acid sequence to fold one way, but another protein that has the same amino acid sequence in part of its structure doesn't fold the same way? So, for example, if we have a protein that has lysine, 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 and it maybe folds into a figure 8, and then we see another protein that also has lysine, lysine, lysine in the same position, but it doesn't fold into a figure 8, it folds into a circle. Why? They have the same amino acid structure. They're both existing in the same cell at the same time under same conditions. Why does one protein fold into a figure 8 with the same amino acid structure and the other protein fold a different way with the same amino acid structure? We really haven't been able to figure this out yet. And what it probably suggests is that there's another layer, there's another level of molecules inside of the cell that are regulating these things that we just haven't found or identified yet.
And is that going to be given a different function to those two different?
It could very well, yes, it could very well. And like I said, with this something we just kind of figured out in the last 15 years that cells talk to each other. And the way that they talk is they secrete tiny pieces of RNA, but they're not messenger RNA. They're weird little snippets of RNA wrapped in a piece of fat. And we call these liposomes. For lipid, they're wrapped in a ball of lipids, so a ball of fat, and inside that ball of fat is this little piece of RNA. The cell membrane, the plasma membrane, is composed of fat. There's a thinking in biology, a term called like dissolves like, and we see this in chemistry as well. If you want to dissolve fat and break down fat molecules, you use other fat molecules. You've been washing and cleaning grease at your kitchen, and you need to clean the grease off your hands. What are you going to use? You're going to use soap. What is soap made of? It's made of fat. So you're taking basically fat and you're rubbing the fat on your hands to get the grease off because like dissolves like. So these liposomes that are these little balls of fat with this piece of weird RNA inside of it is able to, in many times, diffuse right through the cell. It doesn't have to bind to a receptor. It can just diffuse right through the cell. So it gets inside the cell and then releases this weird little piece of RNA that's not big enough to encode for a protein. So what is it doing? We don't really know. We only realized that this was happening about 15 years ago. This is something that we're studying very intensely here, because now that people have looked for this, they find that not only are cells sending out these liposomes with little pieces of RNA inside of them, they're sending out all kinds of little vesicles with different shit inside of it. It's probably used to communicate or to signal or stimulate some response, but we just don't know what it is. So there's all these things that are happening, and this is probably what's happening with protein folding. There's another layer of substructures or submolecules present within the cell that we just haven't identified yet. That's being used to help proteins fold and maintain their shape. And this is one thing that I think we might be able to actually get useful out of Skynet before it puts us all in camps and does the Terminator stuff to us, is that maybe AI will be able to help us kind of deduce this. And this is something that people are very heavily pursuing right now. And trying to find out what that set of rules is that govern how proteins fold has been referred to as the holy grail of bioinformatics. It's kind of like nuclear fission, cold fission. If you can figure out how to make endless amounts of energy, then you're going to be rich, right? So for bioinformatics, if you ever figure out the rules that govern how proteins fold, you'll make $1 trillion.
Is that where epigenetics come in? And maybe the
no,
signal that goes to the cell makes that protein to fold different?
it could be. I mean,
Perception
you know,
is-
there's probably some system that already exists within the cell to help the proteins fold, because if the cell had to rely on a signal coming in, it'd take forever to fold proteins. And what if the cell doesn't get the signal? Then the protein won't fold and the protein can't function. So there's probably a system that's already plugged in inside the cells to help the proteins fold. We just haven't figured out what it is. But it's probably something that's native to the cell, because if the proteins can't fold, then nothing's going to happen. And if you got to wait for a signal to come in, we wouldn't be able to probably function as an organism. I mean, just the fact of us talking and moving around right now, tens of thousands of genes are coming on and off in microseconds. Proteins are being made, proteins being trafficked around. This is how our bodies function. And if you have to wait for things to happen, it'd probably take you 5 minutes to walk across the floor or to pull your pants up or something. So there's probably already a mechanism there within the cell that governs how the proteins fold. We just don't have any understanding of it yet. But this is the stuff that y'all will be able to pursue and figure out over the next 20 or 30 years of your career. So just like we did with the human genome, we're in the process of studying how proteins fold and how they're made. And we're sequencing a bunch of proteins. About 1.3 is, I think, and this was taken from 2016, so the number's probably higher now. Protein sequences are known. They're using every bit of computational power that they can find to look at protein sequences. If anyone plays video games here, they give you an option when you turn on your PlayStation or your Xbox to enroll your console in a protein. I'm trying to think of the name of the project. It's a protein structure project that's related to identifying protein sequences. Somebody figured out a way to take 1 million PlayStations or Xboxes and link them together online and use that computational power to assemble proteins. And so you can opt into this project when you're not playing on your game console. If you leave it turned on, then it will be hooked with other game consoles, and they'll use that processing power to process proteins and fold proteins. So they're trying everything they can to try to process all these different protein sequences to see if they can figure out exactly how they work. So we've got about over 1 million protein sequences that we know, but when we actually look at the structure themselves, we've only looked at about 20,000 of these protein structures. And that's because it's very difficult to actually generate and identify the protein structure in real time. You have to grow them like using X-ray crystallography, which is basically proteins are grown in these very sterile environments, and they're allowed to grow for 2 or 3 years, and they grow like a crystal, like you'd see if you go to a underground cave and you see stalactites coming down from the walls. Those have been formed by millions of years of water dripping on the ceiling and causing the ceiling to slowly erode. And as the water drips, the dirt from the ceiling gets caught in the droplet, and over time you start to see the actual physical projection growing out of the ceiling. A very similar approach is used in X-ray crystallography. The protein that you're trying to grow is basically the mixture squirted at the top of this crystal, and then in a very cold, sterile environment over 6 to 8 months, you'll get a tiny crystal that can be formed, and then you can examine that crystal with a microscope or using lasers to study what the protein structure is. that's the best way that we have to study protein structures right now, and it takes a very long time to generate the crystal, usually about 3 to 5 years to make a full crystal. So it's really slowed down our ability to take the protein sequences that we know and actually translate them into an actual physical structure that we can look at. They've actually been able to, with some recent advances, to get this down from being a year's project to being something that can now be done in maybe months. And it's getting quicker every day. And I would assume that at some point, as we continue to make advances in robotics, that we'll be able to do this now. When I was doing this 30 years ago, you had to do all this shit by hand. It had to be done in a cold room in a very sterile environment. You basically make up your protein mixture, put it in the top of the loading tray. There's this scaffold that comes down. That's a wiring scaffold that allows the droplets of the protein mixture to spin down around the wires, and it just constantly runs down those wires. And over time, because it's very, very cold, you'll start to get a crystal structure that forms. But again, if there's a hurricane and the power goes out, if some dummy goes in there and touches it, if the lights get turned on or turned off, any changes to the environment can completely wreck the crystal and you got to start over. So it's really, really painful and laborious. But they're speeding this up. And one of the ways they're looking to speed this up right now is by doing this with computers and with AI. Again, you really want to be able to kind of handle, I think a lot of us want to be able to hold things in our hand to be able to say, yes, this actually looks like what the computer says it looks like. But this is basically the way we have to study protein structure right now, is using X-ray crystallography. So it's going to take us a very long time. If we got to go from 1.3 million and we've only made 20,000, we've still got about 2.98 million more protein structures to make. If they each take 1 year a piece, you're talking about, you know, 1 million years to make all these protein structures. So we've got to find a way to speed this up. But this is one of the things kind of holding proteomics back to some point. And this is, again, kind of just using a graph to show that we have all these proteins and we've gotten better at learning protein structures over the year, or protein sequences. Once we get to the 90s, we had a dramatic increase in our ability to sequence proteins because of advances in DNA and advances in computing power. If any of y'all were alive in the 90s and you think back to what computers were like in the 90s, the phones that we all carry around now are about 100 times more powerful than the Commodore 64 I was playing on, you know, when I was in the Marines in the 1990s. I mean, so when we saw a dramatic revolution in computing power, we saw a dramatic uptake in our ability to sequence proteins, but we still have not gotten very much better. No big increases in how to make proteins themselves. And so this, until these lines can merge, we're going to struggle with getting all the sequences that we know converted into protein structures that we can examine. And as I said before, this is just something that's going to take us some time to do. People are trying AI and trying other, avenues to try to figure this out. We have been building a database. The United States, the National Institutes of Health have put together databases that are freely available for everybody. And when we finish up the course tomorrow, we'll spend a lot of time looking at some of these databases and some of these resources in general, because there's a lot of this crap that's out there that's free that you can go look at any time that a lot of people just aren't aware of. But there are databases that the government's put out that have the DNA sequence for everything we've identified, every animal, every plant, every gene, and they're doing the same, and it's freely available to the public. You can go download it, and you can get free programs or buy programs that will let you examine the sequence in more detail. But all the data is out there, and they're doing the same thing with proteins as well. So hopefully at some point we'll continue to make advances on this, and we'll figure out what's going on. Well, what are some of the big payoffs from proteomics and bioinformatics? If we get all this data and we can use it to generate these proteins, what are some of the different benefits that we can get from this? I think one of the biggest benefits is that if we can start to understand how these proteins are working at the actual proteomic level, at the protein level, if we know how proteins are working in terms of when they bind to another protein, such as an enzyme binding a substrate, we've kind of worked this part out pretty well. This is something that we've taught classically for about 30 years or so. I think I remember hearing about this when I was in high school in the 80s. So this is something we've known about in terms of how enzymes are proteins. All enzymes are proteins. There is one exception. There's an RNA molecule that's used as an enzyme. But that exception notwithstanding, all enzymes are proteins. And so that means all enzymes are going to have a unique three-dimensional shape that they have to maintain if they want to function. The way that enzymes function is that they have a small region within the enzyme protein that can accept a substrate, shown as this black box in here. So here's our E for our enzyme. The enzyme binds the substrate, and then once you have this binding event, you get some kind of a conformational change in the final protein product, and now this protein is either turned on or turned off, and it will go out and carry out its function. We can also have enzymes that when they bind to a substance will turn them off. We call these inhibitors. So we know how enzymes work pretty well. And so what we're trying to do now is extend that knowledge to other proteins that we've been able to identify to try to study how they interact, how they touch, how they dock when they interact with other proteins. Because again, we always draw this crap as cartoons, so it's easy for us to visualize. But you have to keep in mind when we're thinking about this in reality, you have these two weird three-dimensional shapes that have to come and fit together. And these things are globs. And so they have to find a way for the two globs to be able to come together. And if one glob has something poking out of it that blocks the other glob from interacting with it, then they can't come together. So these proteins have to find a way to interact and then dock and stay together as they carry out their specific functions. And so this is something that we're really trying to understand because, again, the protein shape determines its function. Some proteins are in our bodies doing bad things. And if we can change their shape, then we can change their function and maybe stop some of the bad things that they're doing. A great example of this is the protein that causes mad cow disease that's called a prion protein. So has everyone heard of mad cow disease? If you eat a mad cow burger, then you're going to go crazy. This is the basic premise of this. What this is caused by is a specific protein that normally exists in all mammals called the prion protein. And it has a normal shape that it adopts as it carries out its function. For reasons that we do not understand, this protein can just spontaneously wake up one morning and say, you know what? I'm not wearing my pants on my legs and my shirt on my upper. I'm going to wear shoes on my hands and I put my pants on my head and I'm running around like a lunatic and you can't stop me. This prion protein just spontaneously changes its three-dimensional shape. Once it changes its shape, it takes on a new function and you can't make it change back. Not only can you not make the protein change back, but if it encounters other prion proteins that are in their normal shape, it makes them change into the abnormal shape. When they start to change in this abnormal shape, this protein becomes incredibly tough. It becomes like a Superman protein. You can't break it down. You can't kill it. You can't digest it. You can't get rid of it. And over time, these prion proteins start to accumulate in the body because they can't be broken down. They make their way into your brain and they start to physically clog up your brain cells. And this is what leads to mad cow disease. When you see an animal out in the woods, a deer, rubbing itself against a tree until it bleeds to death, it's because it's got scrapie, which is the term that we use for mad cow disease in deer. So there are proteins out there that once they take on a weird shape, we can't fix them. So if we could figure out how these protein interactions work, we'd have a better chance of maybe stopping some of these bad proteins. Another benefit that comes from understanding how proteins function is that we can make drugs. Again, if you know that this prion protein is going to take on this weird shape that you can't digest it or can't destroy the protein, maybe you could introduce another protein to it that would bind to it that would make it take on a new shape that wouldn't be getting stuck in your brain cells and causing you to go crazy and lose your mind and die when you're 50 years old, which is what we commonly see with people that come down with mad cow disease or one of the diseases very close to it. So drug design is a very important benefit that we can get from understanding how proteins fold. And then the benefit that we, kind of care about to some extent as well, it's not going to get as much funding as drug design or being able to change protein function. That's going to be understanding how everything works, understanding how everything comes together and how things work in the cell. So we get benefits from understanding payoff structure, protein structure in terms of how proteins fold, understanding how, the bread is being made, being able to use that knowledge to make drugs, and then being able to use that knowledge and apply it to maybe other systems in the body so we can educate ourselves and others on how the cells are all functioning and working together. So this is some kind of stuff at the end that you can take a look at for some additional reading. And this concludes this presentation. Any questions on the protein stuff? No?
I had a quick question because you mentioned that there's like a whole bunch of like databases available to look into and do work on proteins.
Yes.
Let's just say like you wanted to come up with your own project and work on it. Like, is this something that you can like be worked into like a publication? Like if you're doing it on your own and made it into a project?
You could, yeah, you absolutely could. And when we meet tomorrow for the session tomorrow, I'm gonna almost exclusively focus on the different resources that NIH has available. Almost all of them are free. I use a lot of them. A lot of us use a lot of these resources because they're free and they're constantly being improved and developed. If you publish a paper, when I publish a paper with a new virus vaccine that we've made, we publish the genetic sequence for that virus and it gets uploaded to the database. So then anyone at any point, tomorrow, next year, 100 years from now, assuming we're still around, can go in and access that data and download it and then freely use it and manipulate it and play with it and see what you're going to get from it. So you absolutely could get a publication if you were able to find or take a protein structure and maybe modify it and model it in some unique way that you could then maybe come up with some explanations as for why it modeled a certain way.
Okay, thank you.
You bet. And again, like I said, tomorrow, I'm probably going to spend 90% of the time tomorrow. We'll probably have a bit shorter session tomorrow, really just focusing on a lot of these resources that are out there because there's a lot of this crap that's there that's been paid for by the taxpayer. We've given grants to different organizations over the years to come up with an easy way to explain to people how DNA functions or how proteins fold, how we replicate DNA. There's a lot of these neat little tools that NIH has that they just have not spent they haven't spent a lot of time telling people that they're out there. But when you find them, they're really easy to use. And like I said, a lot of these tools are good enough to where most of us use these things in our day-to-day applications. And so we'll go over those things tomorrow so y'all can take a look at those and see those things. And we'll also tomorrow when we are going to talk about opportunities that are here at UTRGV as well as around the state and the country for career and for projects and that kind of stuff. So I'll make sure that we touch on those things tomorrow, Christina.
Okay.
Any other questions you're welcome. Any other questions about the lecture before we move on to the second talk? All righty. So the second talk will also thankfully be a little bit shorter talk as well. And let's see, there's something in the chat real quick. Let me see. Okay, we got Christina. Very nice. Okay. All right. So this talk is going to, I can't get to, there we go. We're going to talk a little bit here on the regulation and ethics involved in genetic testing. So I just want to kind of briefly touch on how genetic testing, and this is going to really focus on kind of the commercial aspect of genetic testing. We spent a lot of time in the mini course talking about these things kind of from an academic or educational perspective, because again, this mini course is designed to kind of provide some exposure and to lightly touch on from an educational perspective some of these topics. But there's also a commercial aspect of this as well. And for most people that are involved in science, if you're not in academia, you're going to be using your skills in a commercial setting because that's where the jobs are, that's where the money is. And so understanding how this works from a commercial perspective, I think can be very important. There's a system that's used in the United States, really globally, because what's done in the United States really sets the tone for how things are done in the rest of the world, because we are the largest market, the largest economy. people want to come here and sell their crap, they need to follow our rules. And so when we adopt a structure here for regulation of biological products, blood testing, genetic testing, that kind of stuff, it really gets used around the entirety of the world. So we're going to talk a little bit about what the federal regulations are for genetic testing, how are genetic tests validated, how are they tested, if you will, before they're released to the general public, or before they're used in a clinical setting, kind of how the data is generated, and then we'll spend a couple of slides at the end talking about some of the ethics of genetic testing, particularly kind of circling back to the 23andMe saga that we discussed a little bit over the last couple of days. So when we talk about genetic and genomic testing, this is something that we didn't see a lot of maybe even 10 years ago, but particularly in the last 5 to 8 years, certainly following the COVID pandemic, we've seen a dramatic rise in the demand for genetic testing. So what is a genetic test? A genetic test could cover a lot of things. It could cover karyotype. Let's say maybe your mom has cystic fibrosis. Cystic fibrosis, as we saw from the last talk, is a genetically inheritable disease. And so maybe you want to go in and have your DNA tested to make sure that you don't have the cystic fibrosis gene. And if you do have that gene, then you want to make sure, if you can, and this is going to be dependent upon the person, that maybe you make sure that you don't marry somebody or have a child with somebody that also has a history of cystic fibrosis in their family. One disease in particular that we do a lot of genetic tracking for is a disease called Huntington's disease. I don't know if any of y'all have heard of this or not. It's a very nasty disease. And this is basically a disease that makes you lose your mind when you're about 50 years of age. All of a sudden, out of nowhere, dementia comes on. It usually comes on in late 40s or early 50s. And within 3 to 5 years of the onset of dementia, you're going to be dead. When someone gets dementia, it's not just the fact that the person loses their ability to rationally think. Their brain loses the ability to regulate a lot of the biological functions in your body. You don't know if you're hungry anymore. You don't know if you're sleepy anymore. You don't know if you need to take a drink of water. My grandmother had dementia, and we cared for her the last 6 or 7 years of her life. And sir, I'm sure, assuming there are probably other people in here in the chat that have had relatives that had dementia. The biggest issue that we had is that we couldn't get her to eat or drink because she would think she'd already eaten or drank. And you're like, no, grandmama, you haven't had dinner in like 2 days. You haven't drank water in like a day. No, I just did. But it's that loss of self-coherence and awareness in the moment that puts a lot of dementia people in a precarious position because they forget these basic biological functions, which then, if you're already 85 years old and you haven't eaten for 2 days, that's really bad for you. So genetic testing in cases of Huntington's disease is used to then look at children, because again, you don't see the onset of symptoms until you're 40 years old. Well, most of us have had kids by the time we're 40. So if you have the gene for Huntington's and you don't know it, maybe you marry somebody else that also has the gene for Huntington's and neither one of you know it, just based on Mendelian genetics, right? Gregor Mendel, the Austrian monk that did the pea plant things, everyone remembers that, right? With the Punnett square and how genes are inherited. If 2 people each are carrying a bad gene and they have 4 kids, there's one of those 4 kids is going to have that bad gene, a copy from each parent, which means that bad gene is going to be expressed. So Huntington's is a disease that we try to push people to get genetic testing for, assuming that they know they have a history of Huntington's in their family. If you don't have Huntington's in your family, I would recommend that you not go to the doctor today and say, I need to have my DNA sequenced. Most families are going to be aware that they have Huntington's because, again, this disease comes on in your 40s. That's not normally where we see dementia occurring in people. Usually that's something that we see happening in people generally in their late 70s, early 80s and beyond. So genetic testing for diseases like Huntington has been out there quite a bit. One thing that a lot of us don't understand, though, is that a lot of these tests, a lot of these genetic tests that are out there on the market, genetic tests like the COVID test, if you went and got tested for COVID, that was a genetic test. If you came to my lab when I was testing for COVID 3 years ago, we swabbed the inside of your nose, and then we took it inside the lab. We got the RNA off the nasal swab, and then we cleaned it up and ran it through a PCR and amplified it to see if you had COVID. That test was not officially a regulated test. Because of the pandemic, the FDA came out and gave everybody emergency use authorization, EUA authorization to use the genetic test. A lot of genetic tests that are on the market are not regulated, which means that they get made by a company, they get thrown out there in the market and sold without anybody coming in independently to verify that the test works. For the COVID pandemic, these were all verified in real time because we were having patients come in, we're running the test, we need to get a result in 6 hours. If we're not getting results, then the test isn't working. So a lot of these genetic tests that go out there don't go through any independent analysis to make sure that the test matches what the owner of the test or the maker of the test claims that it can do. So from a patient perspective, this puts both patients and doctors in a really tough spot. I need to do genetic testing to see if you have this disease, but how can I believe the results if I don't know if the test has been, if it actually works, because no one has validated that the test works. So there is, because of this, and this is not something that just started occurring a few years ago. This started to occur in the late 80s. When genetic testing first and PCR first started to gain some traction, big hospitals, big institutions like Harvard and Johns Hopkins started to use genetic testing, and they realized, hey, no one is checking to make sure that these tests work. You wouldn't go buy a car tire from, when you go buy tires at Walmart, those tires have been tested. They've been tested by somebody at the federal government, Firestone, Goodyear, Pirelli, have to submit samples, tire samples to the US Department of Transportation and to the EPA, and those tires are tested. If the tire says it's good for 60,000 miles, then they're going to test to make sure it's good for 60,000 miles. So when you go to Walmart and buy the tire and they say, hey, 60,000 miles, you can be pretty confident that it's going to last that long, approximately, because it's been tested. This isn't happening for genetic testing, which is kind of scary when you think about it. They're spending more time testing tires and baby aprons, which is useful, but than they do testing genetic tests. And so we need to have a mechanism to be able to look at this. So the FDA, CMS, which is the Center for Medicaid, and the Federal Trade Commission got together and basically set up three main criteria, three main things that genetic tests have to satisfy in order to be able to be put out there on the market. And this is analytical validity, clinical validity, and clinical utility. And basically what this means is, does the test work? Does it work in the presence of mutations? And even if the test works, does it mean anything? Does the answer mean anything? Does it give us anything useful? A great example of some of these things we'll talk about, and I'm going to use the COVID pandemic because this is probably something, as far as I know, we were all alive during the COVID pandemic a few years ago. And so this is probably something we can all kind of relate to. So we're going to break these down really quick and kind of talk about these. So the first question that they want to ask is, do you have a way to actually identify what you're testing in a repeatable manner? Does this test even work? One of the common ways that we use for genetic testing nowadays is called qualitative PCR. So we talked a little bit about PCR. PCR is that technique where we can take a tiny piece of DNA and we can amplify it and make a whole bunch of DNA. There's a way that you can do that amplification process if you do it in the presence of certain chemicals, where if you hit the DNA molecule that's being amplified with a laser, it will scatter the laser light. And if you capture that laser light, you can actually count how many DNA molecules are being made. And so this is called an amplification plot. So this is actually an image from a PCR machine that's amplifying DNA molecules. That's what all these different colored lines are. They're each a different DNA molecule, and they're being amplified. And as they're amplified, they're being hit with a laser. They're scattering the laser light. A computer is capturing that laser light and then showing us the little laser light as individual points on these graphs. And so as the machine runs, and down here, this is cycles. So this is basically the running of the machine running. We talked a little bit about cycles yesterday when we talked about PCR, but as the machine runs and amplifies the DNA, you would expect to see the numbers, the amount of DNA begin to increase. And eventually you're going to hit a cap and it's going to plateau off, and you've reached all the DNA that you can make in that little tube. So we can use this growth or amplification plot here to determine whether or not we have a given DNA molecule within here. So during the COVID pandemic, we performed this kind of test. It's called qualitative because it gives us a number, RT-PCR. The RT stands for reverse transcription. What does this mean? When we do PCR, we're going off of a DNA molecule. DNA makes RNA, makes protein. This is the flow of information in biology. We're going to use DNA. Some viruses are not made of DNA, they're made of RNA, and that includes the COVID virus, the SARS-CoV-2 coronavirus. So if you came to my lab 2 years ago and we swabbed you for COVID, or when you went and got tested for COVID, what they're trying to do is get some of the viral RNA, because the COVID virus is made of RNA, not DNA. They're trying to get some of that RNA from your nose to put into the tube. The machine is not testing for RNA, it uses DNA. So you have to convert the RNA into DNA. Remember, the flow of information is DNA makes RNA makes protein. Well, if we're starting at RNA, then we got to go back, or we got to go in reverse back to DNA. So RT-PCR takes RNA, converts it to DNA, and then hits the DNA molecule with lasers and captures the scattered light to make this plot. So as this phenomena is occurring, if you have COVID in your sample, because of some of the chemicals you put in there, you should see this line amplifying. If you put these same chemicals in there and the person doesn't have COVID, you're not going to get this amplification plot. It's just going to basically kind of stay down here in the bottom as a little line. And so this gives you the ability to distinguish between, is this person infected with COVID or not? This gives you specificity. So QRT, qualitative reverse transcription polymerase chain reaction, QRT-PCR, will basically generate a number that you as the technician can then look at and make a decision. Is this a COVID infection or is it something else? It's not COVID, it's something else. And again, when we did this COVID testing, we'd see people coming in. If they had COVID, they'd get these nice big peaks up here. If they didn't have COVID, it'd come in like here, it'd stay down here, maybe a little squiggly line, it would go away. So this is a way by using QRT-PCR to validate your assay and say, hey, what's the output going to be? Well, in this case, it's a number, and people like numbers. You don't want to go to the doctor many times and ask for your blood to be drawn and have your lab values checked and the doctor not tell you what those numbers were, right? You go to get your blood pressure done. You want to know what the blood pressure is. You don't, I mean, right? Most of us do. You don't just want to hear, yeah, it's good. You want to, hey, well, what was it? Oh, it's 110/70. We like numbers. We all like numbers because for us, it lets us put things in a certain box. It lets us qualify certain things. The same thing is true for genetic testing. So if you use RT-PCR, you can get a number, not just a yes or no answer. And numbers are what doctors and physicians like to look at when they're going to make a diagnosis. So the first step in a new genetic assay is making sure that the assay works, and then it gives you some kind of an output that you can measure. The second question we want to ask is, okay, and we did this during the COVID pandemic. Remember, we had all these mutants come out, right? We started with COVID, and then we had Delta, and then we had Gamma, and then we had Omicron, and then we had Theta, and then we had monkey butt juice. I mean, had all these weird COVID variants. And so when you went in to get tested, the question was, can the test that was originally designed to identify regular COVID identify these different mutants? And what we saw during the COVID pandemic is that many of the assays that were being used to generate this data didn't work if you were infected with a mutant. Now, the test that we used did work to identify mutants. We couldn't tell what mutant it was, but we could tell whether or not you were infected with a mutant form of COVID. Okay, you can tell whether or not I'm infected with a mutant. Does that matter? Does it mean anything? And in the case of the COVID pandemic, it didn't. We thought that it would. Right in the beginning, they were like, oh, you got Delta. You have to like stay home extra long time, or you can't go anywhere, not even the store. I mean, and what we realized as the pandemic went on and we saw all these mutations, these mutant strains of COVID arising, that it didn't make a shit. Didn't make one difference whether you had Theta, Delta, Omicron. The disease was pretty much the same. It was pretty much going to be the same. And being able to test for and identify the different mutants wasn't going to affect your treatment options. If you came down with Omicron, they don't care that you have Omicron. They care that you have COVID and you're going to get treated for COVID, because we saw that differences between the different COVID mutants didn't really have an effect on the outcome of the disease. And we didn't have different drugs that would only treat Omicron or only treat Theta or only treat Delta. So even if you can develop a test that can identify what the organism or the pathogen is, if the pathogen mutates, does your test still work? And even if it does work, does it matter? Does the answer matter? And again, for the COVID pandemic, we didn't really care. We were able to identify whether or not people had mutants. And I would have people call the lab and say, hey, I got my COVID results. What does this mean? And I'd say, well, it means that you're infected with COVID and you have one of the mutants. Which one? I don't know. You have a mutant, but don't worry. They're going to do the same. They're going to beat you with the same stick, whether you got a mutant or you have the wild type version. So it doesn't matter. There are some diseases, though, where being able to differentiate between different strains is critically important. Anthrax is a great example. There are 2 strains of anthrax in the world. There's the strain that kills you in a day or 2, and there's the strain that won't hurt you at all. Those 2 strains, the lethal strain is called Ames, and the vaccine strain is called Stern. We actually use the non-lethal form of anthrax to vaccinate cows and sheep and goats and all the animals that are out there grazing, eating grass, because anthrax naturally occurs. There's anthrax all over Texas. I could walk out here right now and probably spend 5 minutes digging in the dirt and find some anthrax out there. It's not going to be the lethal version. It's going to be the vaccine strain. But if you go to get tested for anthrax, you'd like to know, do I have the version that's going to kill me in 2 days, or do I have the vaccine strain? When we had the anthrax attacks, and I don't know, many of y'all may not have been alive during this, but right after 9/11 in 2002, some asshole sent anthrax spores through the mail, and it killed about 14 people and made about 100 people sick. We had to shut down the mail service. I mean, it was a nightmare. This is where we got the whole thing about, now we see, dummies sending envelopes with white powder in them. That's where that came from, because the anthrax spores, when they were sent through the mail, they were lyophilized. They were dried as a dry powder. That's one of the easiest ways to spread anthrax spores. And then when we actually deploy anthrax spores as a bioweapon, that's how we do it. We take a dry powder, we mix it with some water, and we shoot it out a sprayer that can disperse and spray the spores over a very wide area. So those people needed to know, do I have Ames or do I have Stern? If I got Stern, I can go home. If I got Ames, I need to get to the funeral parlor because I got to get my affairs in order. So tuberculosis is the same way. There's a strain of tuberculosis that we use as a vaccine, and there's a strain that will kill you in a couple of days. So sometimes you do need to be able to identify the strains, but for COVID, it didn't matter. So whether or not a genetic test can differentiate between mutants and whether that even has any meaning is something that needs to be addressed when you put out a new assay. And then the last question that we ask is, does the result even matter? Okay, you could tell me that I have COVID. You can't tell me whether it's a mutant, but you can tell me that I have COVID. What use is that information? Well, if you have COVID, we can tell you, hey, like we did during the pandemic, you need to stay home for 7 days. You need to stay out of public areas. Try to limit your interactions with other people. And during a pandemic, if you can rapidly identify people that are sick, then this gives you an opportunity to intervene before they have the ability to spread the infection, such as, hey, stay home for 7 days. And this can be beneficial. Or maybe you get identified with COVID, you get tested positive for COVID, and then you can be assessed and they can say, hey, your blood pressure is dropping, your HRV volume in your heart is irregular. Maybe we need to admit you. So there are going to be examples where getting that answer is going to be meaningful. There are also going to be times when you may use a genetic test and it gives you an answer that doesn't really do anything for you. Let's use influenza as an example, or even to some extent the COVID pandemic. Okay, I'm infected with COVID. What do I do now? Oh, well, you go home and stay home. Okay, what else? That's it. Stay home and good luck, and hopefully we'll see you in a week. Sometimes the answer from the genetic test may not be any benefit. It may not lead to an improvement of the health outcomes. And if you're not going to get an improved health outcome, then what's the purpose of even doing the testing? So whether or not the test works, whether or not the test can work in the presence of mutants, if there's mutants in the testing population from the pathogen's perspective, and whether or not the answer even means anything, are the three big questions that you need to ask. And so in order for the federal government to get their arms around this problem, the government has set up something that's called CLIA. And CLIA stands for Clinical Laboratory Improvement Amendments. And there's a law that was passed in 1992. The law was written in 1988. It passed in 1992. It's called CLIA 88. And this is basically a set of standards that the federal government has put in place for all commercial licensed testing labs in the country. And this has really been spread across the world. These are standards that you must meet in order to be able to accept and test and release patient samples and results, and to be able to bill patients and insurances and physicians for the testing. And so CLIA comes down to four things. Laboratories must be certified. This certification is done at the state level. The labs have to all meet very high standards for quality, and they tell you the standards for how you operate the lab, the standards for the personnel, the people that work in your lab, the kind of quality control that you use in the lab, and how you make sure that your testing and the staff that are doing the testing are doing it properly. CLIA takes the testing, and this is all testing done. So you go to the doctor's office to get a glucose test, it's got to meet CLIA regulations. You come to me to have a COVID test done, it's got to meet CLIA regulations. CLIA covers all testing done by labs on patient samples. If you get caught doing testing on patient samples and you don't have CLIA certificate, not only are you going to jail for a long time, but you're going to go broke. And the company or place that you worked at is going to go broke as well. CLIA can fine you up to $100,000 for each mistake that you make. And just like the way law enforcement works, the cops can really pull you over for anything. There was a judge 2 years ago in McKinney up in North Dallas. They got pulled over. He'd been fighting with the sheriff's office there for like 10 years. He and the sheriff hate each other. And they caught this judge one night and they thought he was drunk, but they couldn't tell. They needed a reason to pull him over. They pulled him over because the police officer said that the tire tread on his tires was too low. How do you see tire tread at 80 miles an hour in the dark when the car is moving? I don't know, but they used that to pull this judge over. So they can basically have wide latitude in levying fines against you from the police perspective, and CLIA can do the same thing if they think you're messing up. So, but they've organized the testing based on complexity. Either it's a test that's waived, which means many times at the doctor's office, if you go in to get a blood test or something, they have bought a machine that has been designed and constructed to where it doesn't need to have specific training or specific qualifications to run the test, and CLIA considers this to be a waived test. From a billing perspective, you can't charge as much for a waived test as you could for a regular test. So if you come to my lab and say, I want a glucose test. Normally, I'm gonna run that on my regular chemistry machine and it's gonna cost you $20. If you go to the doctor's office, they may be able to run a very similar glucose test, not gonna give you quite as much information, but it'll cost you $5 because it's a waived test. So there are waived tests, there are tests that are considered to be moderately complex. This is gonna be chemistry, hematology, and then tests that are considered to be high complexity. That's gonna be pathology and genetic testing. And then the personnel and lab requirements for each category of testing is going to be different. And then lastly, all of this crap is enforced by CLIA coming every year and inspecting your lab. And so what these certificates are over here are the certificates that we have for our CLIA lab here in the Department of Human Genetics. And so we registered the lab. Once you register the lab, they give you a little certificate. You have to submit a bunch of paperwork saying, this is who's in charge of the lab. These are what my qualifications are. They have a very specific set of qualifications that you have to meet in order to be the director of the lab. They're based on your education and your experience, not only experience working, but experience supervising other people carrying out lab testing. And then once they come and you pass your inspection, they upgrade your certificate to say you're now compliant. And then on the back of this, which I don't show, they actually list all the different testing that your lab is allowed to do. So CLIA is used to basically enforce how testing is done, particularly genetic testing, across the entire nation. And again, if they catch you making mistakes, they will charge you fines. And if you get up here to where they just decide that you're basically just being a shithead and you're making mistakes on purpose because you don't care, up to $2 million per violation. And labs get shut down all the time for this because not only will they beat you to death with these fines, but they will also make you reimburse the fees that you charged every patient that came in and got tested in your lab while you were committing these infractions. And then you got to send out letters saying, boy, I suck to all these people. Basically, this will kill your lab. So when CLIA comes to inspect, they don't come to inspect with the hammer. They're coming to help you. But if they find deficiencies that are too severe for them to help you, or if they find problems and they give you a chance to correct them and you don't correct them, they can come down and completely shut down your lab. And it's very expensive to get a lab started. And once you lose a certificate, you will not get it back. And again, they have put people in jail for violating CLIA. They put people in jail all the time for breaking CLIA because the government's role is to try to make sure that patients have and physicians have confidence in the lab testing system when they go to get tested at a clinical lab. And so they want to make sure that people know what's going on. So yes.
So let's say CLIA comes down and since you're the director, but it's someone under you that's doing the mistake, do they find that person at the
No.
lab?
They find the boss.
So they find you?
Well, they find the lab and then any penalties are going to be levied against the director of the lab. Because at the end of the day, I'm responsible for what happens in the lab. It's my name on the certificate. So it's just like if I have students or staff working in my lab and one of them sets a fire, the first person they're going to come look at is me. How did this happen? What are you doing in your lab to allow such a thing to occur? And CLIA takes the same perspective. The amount of paperwork that you have to generate to have a lab function to satisfy CLIA is insane. I mean, we have policies that tell you, describe how employees are going to clean machines, how many times a week they're going to clean machines. Where are they going to write down that they clean the machine? Is someone going to check to make sure that they clean the machine? And then that person has to document that they checked that the machine's being cleaned. Has someone checked the temperatures in the fridges every day? What about the temperature in the room? What about the temperature of the incubators? Are we making sure that the materials being used are not expired? What's the lot number? What's the expiration date? Everything has to be documented. There's a very common saying in the regulatory environment, if it's not written down, it didn't happen. If you have gaps in your record keeping where you didn't take the temperatures for 6 months because Joe Blow forgot and you didn't tell him, they're not going to get mad at Joe Blow. They're going to get mad at you. You're going to be the one that gets fired. You're going to be the one that gets fined and penalized. So running a lab like this is very, very serious. And the way that I make it happen is because I can't possibly know all of this shit. There's just too much to know. So what I do is I find good people that I trust that do know these things, and then I put them in charge. So when I ran the COVID lab, I found a lady to manage the lab who had been doing this CLIA work for 30 years. She knew the regulations by heart. This is like a 400-page document. She knew almost the entire thing by heart. And so my superpower was being smart enough to recognize that she knew more about this than I did. And so what I did is I hired her, put her in charge of stuff, and then we would sit down and she would review with me, hey, I had to do this. Somebody didn't check this. Oscar forgot to write this down. And she put in place a very good system to track everything, but everything has to be tracked. When samples come into the lab, how are the samples transported to the lab? Did the courier keep the sample maintained at the proper temperature? Well, how do you know? Because you got a thermometer in there and you're logging the temperature. I mean, the amount of paperwork required to make these things go is insane. Once you put the system in place, it'll kind of run itself, but it's very, very easy to screw this up. And a lot of people do because they don't take the time to go through all the regulations and they just say, I can wing it. And then CLIA shows up and they start crawling up every orifice of the lab and you're like, oh my God, you're not allowed to have things under the sink. The sink has to be clean underneath the sink. Those little cabinets, it can't be shit under there. Not a dustpan, not a bottle of bleach, nothing. They have very specific requirements. And if you don't meet those requirements, they will beat you down. So the same kind of oversight that they use for genetic testing is just as intense. There are about 200,000 labs that are enrolled in CLIA across the United States. Only 600 of those labs do genetic testing. So when you think about that, I mean, there's probably 10 CLIA labs in Texas. Texas, pretty big place that do genetic testing. I mean, genetic testing is not done by a lot of labs. And you're required to share all of your data, all of your information with CLIA so they can track and make sure that things are being done properly. And again, the requirements for genetic testing are very, very high. You have to make sure all of this stuff is being not only checked, but documented that it was checked on a daily basis. And if something doesn't work, then you have to explain why it didn't work and what steps you took to correct it. If something didn't get written down, then you're required to figure out why it didn't get written down and then provide an explanation for it. You have to make the staff do daily testing of all the different assays that you run. They have to be tested we did it on a weekly basis to make sure that the test is functioning properly. And what I would often do is I would have different technicians come in and test each other's samples to make sure that we're all getting the same result. Because sometimes some people have magic hands and some people have hot dog fingers, and you'd have people come in and they would do the testing, and sometimes the results would work and sometimes the test would fail. And then I have my backup crew that I knew were good, and I'd have them come in and run the same test and see if they got the same result. And if they got different results, then I'd pull those two people out and say, okay, one of y'all is a shithead. Let's figure out what's going on. Did someone not follow the process? Did someone forget about something? And you'd figure out, okay, somebody saw something that said 200 and they thought it said 20, and they're not adding the right volume of a certain chemical. But all this monitoring has to be done. For all other types of testing in labs like chemistry and hematology, this quality control, this QC, is done on the machine. So the technician doesn't have to mess with it because it's an automatic feature built into the machine. That doesn't happen for genetic testing. And that's why the requirements for the staff for genetic testing, the qualifications are very, very high. Generally, to be able to do genetic testing, to do any kind of testing, you have to have at least a high school diploma. In Texas is one of 12 states that does not require you to have a medical technology or a medical lab technician degree to be a med tech to do genetic testing. If you have a high school degree and you've been properly trained and all that training has been documented and you've been evaluated successfully and shown that you can do the test, then you can run it. For genetic testing, you have to have at least an associate's degree and at least 1 year of experience working with this test before. Your 1 year can't be the first year of work. It has to be before then. So one of the reasons that my lab was assigned to do the COVID work is that we were one of the only labs on the UTRGV campus 5 years ago that were doing genetic testing. We were doing genetic testing of mosquitoes collected all over the valley looking for Zika and dengue virus. And so my graduate students had been doing RT-PCR on about 4 million mosquitoes. We did about 1 billion mosquitoes over 4 years. And so everybody in my lab had been doing this shit for years, and we had published half a dozen papers on it. So they had proof. And when we applied for the CLIA license, I had to submit those papers in to CLIA to show, hey, Oscar and Juan and Marisol and Dion, all these people that are going to be the technicians, they've been doing this. Here's the proof. There's a paper we published in 2015 and 2017 showing that they can do this stuff. The requirement for doing genetic testing for the staff is extremely high, much higher than everything else. And there's some additional regulations that you have to meet as well. And a lot of this stuff is stuff that's, is very laborious. I mean, you have to enroll in a testing program, which means that twice a year, we have a company that sends us samples. They're unknown. We don't know what they are. And they say, here's your samples. And we're supposed to figure out what they are, test them, get the result, and then send them back. So it's called proficiency testing. And we get this done every quarter. So 4 times a year, we get samples sent from API, American Proficiency Institute, that we're supposed to test. And we have to get like 95% or higher. And if the result comes back wrong, then I got to retest it. I got to pull that technician out and make sure they're retrained and do all this kind of stuff. So the standard is very, very high. Everything has to be tracked. You've got to maintain patient confidentiality. The specimen has to be maintained. When we were doing the COVID pandemic, we were saving shit for a month because we'd do a test and then a week later, we'd get a call from the physician or 2 days later saying, hey, this person, are you sure they have COVID? Are you sure they don't have COVID? This and that. We'd have to go back and retest. And this happens for all testing. So you've got to maintain all these specimens. Well, they got to be saved and stored in a place that's going to keep the specimen intact. You've got to document who's going in the room, who's going out of the room. So you know everything's got to be barcoded, logged. If people call and complain, I don't like the way you swab my nose. Joe was mean to me. All these things have to be tracked. We had to do quarterly surveys, right? Send out surveys to patients and physicians. Are you happy with us? Do you like us? Do you not like us? What did we do good? What did we do bad? I mean, it's a massive amount of paperwork that has to be, and a massive system that has to be generated to be able to track samples. And for genetic testing, it's all ramped up to 1,000. So there's a lot of stuff that you have to do to make sure that you meet the CLIA requirements for genetic testing. And then, as I said, there's very specific requirements for the personnel. The lab director is going to be overall responsible for everything. And then everyone's going to have a separate role that they play. We'll have a separate MD to be the clinical consultant. You could maybe have a technical supervisor or a general supervisor. You're going to have your testing personnel. And then we did it every 6 months, but at least every year, everybody that's doing testing has to be verified and tested to make sure that they're competent. And the highest parameters and highest level of qualifications is applied to genetic testing labs. So this is a tough thing to get set up. And unfortunately, unlike chemistry and hematology testing, you can't go to school to learn how to do genetic testing. There's no class at UTRGV or UT Austin or Harvard that teaches you how to do PCR or how to do RT-PCR. The only way that you learn how to do that crap is you get into a lab that does it for a living, like my lab does. And so the students that came into my lab, that's how they learned how to do genetic testing. And so when they came to me and said, hey, we need you to start this COVID pandemic thing, that's why I told them, look, you got to hire everybody in the lab because I can't go find these people out in public. There's not a class that people are taking up here that teaches them how to do this. They have to learn this in a research environment more than likely, and it's hard to do. And again, there's just a lot of stuff that you've got to be responsible for when you're doing genetic testing. Everything's got to work. And look, they understand that you're not going to be perfect, but what they want to know is that when you make a mistake, what are you doing? Are you hiding the mistake? Are you sweeping that shit under the rug so nobody looks at it? Or are you confronting the mistake and saying, okay, we screwed up. What can we do to make sure this doesn't happen again? As long as you are not hiding from your mistakes and you are making an honest attempt to address the mistake. So for example, we were having issues where we would have clinics leave samples in the box outside the clinic, the little boxes that are outside the clinics, and then we'd have a courier go by and pick up the samples. And we had a schedule, and the courier would go by at 5 o'clock, the clinic closed at 4:30. Courier goes by at 5 o'clock and grabs samples out of the box. And then the next morning, the courier goes over there, and guess what? There's freaking samples in the box. Somebody in the clinic, after the courier left, would go out there and drop some samples in the box. Well, they sat there in the heat in July overnight. Sometimes they did this on a Friday, and the courier would get them until maybe Sunday. And then the samples sat there for 2 days. And then I'm saying, why are you doing this? And they're like, they were in the box the entire time. The courier's an idiot. I had to go buy stupid cameras, like the body cams, for the couriers. So the courier, when he walked up to the box, I could see the box, I could see what he got out of the box, and the courier is also logging everything down. He'd scan it with a barcode scanner and scan the samples so we would know what was in the box. I had to do this shit so I could go back and tell the physician that ran that clinic, no, dummy, one of your staff went out there at 5:30 before they ran home and dropped the samples in the box. I know because we have body cam footage showing what the guy got out of there and showing the log. But this kind of stuff happens. And so as long as you're making an attempt to address the problem, CLIA is gonna go with you and they're gonna give you some space. The last little thing I kind of want to mention about this before we wrap up, and I know we're just about here to the end, is, and a lot of us probably saw this during the pandemic. So the last year or so of the pandemic, we all started to get these at-home COVID testing kits. Do y'all remember these? I actually still have a couple of these things at home because my wife, every time she gets a cold, she's convinced she's got the coof. So she's like, I gotta get tested. And I'm like, I'm not testing for COVID anymore. Go get the damn kit from China and use that damn thing in there, you know. But these kits are referred to as direct-to-consumer genetic testing kits. And one of the things that we've been seeing, and if you notice, this lines up very nicely with what we saw with the COVID pandemic. Prior to 2020, you didn't really see this. Outside of 23andMe, you didn't see genetic testing kits being used as a direct-to-consumer means that there's no middleman, there's no lab or physician involved. You get the kit sent directly to you, you do something with it, and then you send in the sample and you get back your result. So for 23andMe, it was spit in the cup until you get this much spit in there and then wrap it up and send it in and we'll sequence the DNA. For the COVID direct-to-consumer testings, the kits that I got, that most of y'all probably have, is some swabs, and you swab yourself and then you put it in a little tube and you mix some shit in there, and then after you shake it up, and then you take the little test strip and you put 3 drops here and 3 drops here and wait to see if you get the lines. It's just like the pregnancy test, right? For guys, we don't maybe know, but if the ladies have done the home pregnancy test, it's the same thing. Well, this is something that the testing market is really leaning into, because this makes it a lot easier for people to do a genetic test at home. And then if they get an answer that they don't like or that they're not sure about, then they can go to the physician or to a lab and get a follow-up test. So in 2024, this market was about $2 billion. Look at what it's going to grow to in 10 years. It's going to almost increase by a factor of 5. So this direct-to-consumer genetic testing is going to be something that's going to increase to be very much a very lucrative business. This is kind of like when Amazon came on. And now everybody buys everything from Amazon because you can get whatever you want pretty cheap, and you're going to get it sent to your house in a couple days. It's a lot easier than me driving around. I got a new phone a couple days ago because I smashed my iPhone a few months back. And so I got the new phone and I'm like, I need to get a case. I forgot to order a case. What's easier for me to do? Search at Walmart and then go to the Walmart, and maybe they say they have it online, but they don't have it, and go to Target and do this, or order it on Amazon and I get the case by tomorrow morning. So it's so much more convenient, right? To do it kind of where you get the kit and you can do it. And so the same thing is being done with genetic testing. And so as we move through the next few years, this take-home genetic test is going to get very popular. It'll be very nice to be able to get the result quickly. And then, like I said, if you get a result that you're not sure about, you can always then go to a proper physician, to your doctor, to a testing lab, and they can give you a confirmation check. CLIA has done some basic analysis of genetic testing, and that's kind of what these bullet points over here. And basically, GT stands for genetic testing, and this is just kind of CLIA's general feelings on genetic testing. There's no widely accepted definition of what a genetic test could be. The COVID take-home test is considered a genetic test, just like you coming into my lab and me doing an RT-PCR analysis on your sample is considered a genetic test. Those are wildly different. One of them is using PCR and amplifying DNA from RNA. The other one is you sticking a Q-tip up your nose, mixing it with some salt water and squirting it on a piece of paper. So that's two widely different definitions of what a genetic test is. But basically, as far as the government is concerned, they're okay with seeing this direct-to-consumer market go. And from the government's perspective, they think that genetic testing is kind of working okay. So there are some things, and we're going to spend the last 2 minutes here. I know we're done here, but I want to get these last two slides. There is one thing that I do want to talk about for genetic testing, and this is genetic discrimination. And this is something that we didn't really consider before we got to the point that we're at now, where we now have the ability to sequence DNA. And what is the big concern about genetic testing? Well, if you have genetic information, I have your DNA sequence. I can look at your DNA sequence and I can say, wow, Natalie looks like she has a history of high blood pressure and diabetes and obesity in her family. She doesn't have high blood pressure or diabetes or obesity, but there's a history of it in her family because she has some of these genes for it. So as the insurance company, we're going to charge you an extra $1,000 a year because at some point you may get diabetes, you may become fat, and you may get sick, and we don't want to be covering that. And so we're going to charge you extra money. And so this is a very big concern, right? We're going to get genetic testing done because you want to know maybe you got a disease gene that you didn't know that you had. Maybe you have Huntington's and you have a history, or somebody in your family might have had it. My wife has an aunt that has Down syndrome. And so we started our family late in life. And so when we started having kids, that was one of the things we were concerned about. I said, hey, once a woman gets past the age of about 32, 33, the chances every year beyond that age for the child to be born with Down syndrome goes up by about 1%. And so that was something we were concerned about. And so she went and had genetic testing done just to make sure everything was okay. Well, if the insurance company or somebody gets a hold of that data, they could use that data against you. They could say, oh, well, your genetic data says that your left foot is shorter than your right foot by 1/2 inch. And so maybe that will affect how well you can stop your car. We need to charge you more money for car insurance. This is something that's driven a lot of concern at the federal government level. And so a law was passed in 2008 that's called GINA, which is the Genetic Information Non-Discrimination Act. And this is basically a law that says you can't use people's genetic information to discriminate against them. So it sounds good and it's a good start, but there are some caveats about GINA. So GINA basically says in part 1, you cannot use genetic information to discriminate against people with regards to giving them health insurance or charging for health insurance. And you also can't use their genetic information to discriminate against them in hiring. So let's say you come to me to get hired and I have your DNA and I look at it and say, man, this person is healthy, but they have this gene, which if they have a child, their child might be born sick. And then I would have to pay the health insurance for that child because they're an employee. I don't want to hire this person. Or, oh, this person has a gene which makes them more likely to become an alcoholic than this person over here. So, well, I don't want to hire this person because even though they're not an alcoholic, maybe in 10 years they'll turn into one. So GINA makes it illegal for you to use genetic information to discriminate against people, but there's a catch. If you are a small company, if your boss or your company has fewer than 15 employees, then they can use the information to discriminate against you. Huh? I don't know how they made that cutoff. I'm sure this is something that the politicians did based on money and shit, but Gina doesn't apply if your employer has fewer than 15 employees. And it also doesn't protect you against being discriminated against for any other type of health insurance except health insurance. So life insurance, disability, long-term care, your health insurance can be used to discriminate against you. So they look at your DNA and say, oh, they get a hold of your DNA and say, oh, Natalie might get diabetes. She doesn't have diabetes now, but that's because she's 25 and she's healthy. Maybe when she's 45 and she's worked here for 20 years, maybe she'll get diabetes. Well, okay, we're not going to give her long-term care insurance, or we're not going to give her life insurance because she may be diabetic and we may have to pay that out earlier. We'll give her health insurance, but not any of these other insurances. So Gina is a nice start, but it's not really going to do anything moving forward. But this is something that the government is aware of, that they're trying to address. This is the last slide that we're going to talk about here for the day, and this has to deal with epigenetic discrimination. You kind of mentioned a little bit about this, Raul, a couple days ago, and this is really what is at the heart of the 23andMe fiasco, which is kind of mentioned down here. So epigenetics is the study of changes in gene function that are inheritable, that can come from your cells dividing, that doesn't involve a change in the DNA sequence. So you don't have anything in your base DNA sequence that is going to lead to something bad, but maybe at some point your cells will divide, you'll pick up a mutation. This is pie in the sky crap, and this mutation will then make you sick. And so there is a way, by looking at some of the characteristics of DNA, that you can use DNA genetic testing to predict some things, such as how people will respond to exposure, maybe how they will respond to certain chemical treatments. This can also give you information about what they've been exposed to in the past. This is a topic that we're looking at right now here in the Department of Human Genetics. We have a lot of DNA sequences from a lot of people all over the world, but particularly here in South Texas. And we're looking to see, hey, how many times have these people in the past been exposed to toxic levels of pesticide? Do y'all know that the largest oil spill and EPA disaster in Texas is here on the Brownsville campus? That gas station, that Valero station that's right over there, in the 1960s, underneath that thing is a massive chemical spill. Massive chemical spill, the largest in Texas. Yeah. Just right out off the campus where that gas station is. The largest spill in Texas, the largest chemical spill in Texas. So people been living here 50, 60 years. That spill happened I think it happened in 1958 or 61. So over 50 years, 60 years, right? Almost 70 years. So you've had a couple generations rise and fall in this environment. And so one of the things we're looking at is to see, hey, any people that have been around this area, are they displaying in their DNA exposure to toxic pollutants or other factors? And so these things, even things like stress, social issues, social adversity, have been shown to affect DNA. And so the concern about this is that 23andMe has sequenced a ton of DNA. They have all this data. Now they've gone out of business and they're bankrupt. The only thing that's worth anything in that company is that genetic data. No one cares for their chairs or the tables in the building, or their, they probably didn't even sequence the DNA. They probably sent it to another lab that sequenced it and then gave them the data. But they have all this data. It's worth trillions of dollars. Of course, if you're the owner of the company, you want to sell that shit, because not only do you want to walk away with some money, but you owe a bunch of money to all the people that invested in 23andMe when it went public as a stock. So you got to pay those people back so you don't go to jail, and you'd like to have some billions to ride off in the sunset. So the company is trying to sell this data, and people are pushing back on it, because what they have determined during the initial steps of the lawsuit is that 23andMe has been selling off data from people that are Jewish and Chinese to third parties for unknown reasons. One of the things that is concerning about this is that We developed the technology in 1992 to genetically use genetic information to target weapons. What do I mean by that? We can design a bomb that will release a biological agent, a virus that we can target to only affect Hispanics, to only affect Muslims, to only affect Jewish people or Chinese people, because we all have unique genetic markers that are tied to our ethnic heritage. There was a dumbass in 1996 that took tobacco mosaic virus and modified it to kill marijuana plants. And his genius idea was to release this virus all over the country to kill marijuana plants. It's like, dude, did you not see that Will Smith movie, I Am Legend, where they made a virus to kill cancer cells and it wiped out mankind? If you make a virus and release it to kill marijuana plants, what is your guarantee that it's not going to kill all the other plants as well? Because mutations happen. There's no way to control that. The same thing can apply to these bio-targeting weapons. If you design a bomb to release a virus that's only going to target Jewish people because it's targeting specific genes that are more prevalent in the Jewish community or in the Chinese community or in the Hispanic community than other ethnic populations, what is your guarantee that that thing is not going to change and start killing everybody? And so the usage and sharing of this data has become very, very contentious. And and the company is saying that hackers got it. It was the hackers. They got access to it. And then it was the hackers that sold this data. But this question, the simple fact is that 23andMe has organized its DNA data by ethnicity. That's very odd. What we organize samples in our lab by time. So I know what came in 2020, 2021, 2022, 23, 24. If I want to look at all the genetic data, I can go do that, but that's because it's genetic testing. This is chemistry testing. This is hematology. This is microbiology. These people are organizing their genetic data based on specific ethnicity. And not that that necessarily is a bad thing, but if you do get hacked, people can then take this data and sell this to people that have bad intentions that may want to design a bomb that can go out there and wipe out all the Chinese people living in a certain place or all the Jewish people living in a certain place. And so what happens with this data from 23andMe is going to be very, very critical to see moving forward, because there's a lot of bad stuff that can come from this and not a lot of good stuff. In my opinion, the only thing that they could really do is give this data to the government, because the data is already out there. There's no way that you can put the GD back in the bottom. The best thing you can do is give it to somebody that you, and again, we're saying the government, so I have to say this and bite my tongue, that might be best equipped to protect that data, because they're not going to destroy it. There's no way that they'll destroy
And the
it.
government sees that information.
Right now they can't know because of bankruptcy laws and stuff. And if the government goes in, and because you could make the argument if you're an attorney, well, if the government can go in and seize 23andMe's data, their personal assets, then anytime a company goes bankrupt, they can go in and seize all their assets. Well, that's not going to work in a capitalist marketplace economy. People won't want to do business here. That's why no one wants to go open up new businesses in New York, because they went after Trump for $500 million for BS. And other people that own companies see that and they're like, I don't want to open up a new company in New York. That guy, Kevin Leary from the Shark Tank, is trying to build a $10 billion data center, a massive server farm to handle massive amounts of computer data for crypto and for DNA sequencing. And he was going to build it in New York because they have the Niagara Falls power plant. He was going to build this thing in New York. He moved the data center to Nebraska or to North Dakota instead, because he saw what happened with Trump and said, well, what if they come in and do that to me? If they can do this to Trump, they can do it to anybody. And so the same thing kind of applies with some of this stuff. And that's why you won't see the government probably come in and try to seize the data. But I think the best thing to do would be for the government to maybe buy the data. And then they at least somebody can control it, maybe have we can have maybe a tiny bit more confidence than where we're at now. But this entire usage of genetic data to discriminate against people based on their past exposure or what kind of past information you can get from their DNA is referred to as epigenetic discrimination. So that's kind of my final thoughts on this. I'm sorry, I know we ran a little bit over today, about 17 minutes over. Any questions before we wrap up? No, no questions? Okay, guys, well, thank you so much for attending. Tomorrow will be the final session, and we're primarily going to just focus on looking at some of the different resources that are available. And then I want to spend time talking about educational and career opportunities that we have not only here within the Department of Human Genetics, but at UTRGV, at the School of Medicine, and kind of what I see happening over the next 10 years or so as we continue to see the economic environment dramatically change here in the Valley. So hopefully we'll see everybody tomorrow for the final session. And then after we wrap up the session tomorrow, I'll probably, and I'll talk more about this tomorrow, I'll look to be giving out everybody's certificates and sharing all the slides that we used in the next few days. So thanks again for attending, everybody, and I look forward to seeing you all tomorrow.